Photosynthesis. It is the process by which green plants convert light energy into chemical energy in the form of sugar. This is what we see in the above diagram. Light energy hits the leaf, and the leaf is therefore able to synthesize sugar, primarily glucose, from the process of photosynthesis. There are things called pigments in the plant, which allows light energy to be absorbed, and they are called photosynthetic pigments. The main photosynthetic pigments are chlorophyll A and B, and they absorb blue and red light. There are also carotenoids, which are termed accessory pigments. Essentially, they absorb light that is not absorbed by the chlorophyll pigments. In other words, the carotenoids extend the range of light being absorbed. Now let's actually get into the photosynthesis. It is a two-stage process, and here we will look at the first stage, commonly called the light-dependent phase. In this initial stage, the light that is incident on the plant gives energy to the electrons within the pigment molecules. It is commonly said that the electrons become excited. These electrons can then pass through a transport chain, where the energy molecule ATP is produced. Energy is also used for photolysis, meaning the splitting of water. In photolysis, the H2O molecule is broken down into oxygen, which is released as a byproduct. The hydrogen ions left are picked up by a coenzyme called NADP, forming NADPH. This NADPH is seen in the second stage of photosynthesis, as it carries the hydrogen ions to the latter stage, allowing the production of glucose. The second stage of photosynthesis is called the carbon fixation stage, and is the process in which the sugar glucose is produced. We start off with a substance called RUBP. An enzyme called RuBis cofixes carbon dioxide to the RUBP, removing it from our atmosphere. This fixation of CO2 to RUBP forms another substance called 3PG. This 3PG is then converted to G3P, another substance. This occurs as 3PG is phosphorylated using ATP. Phosphorylation simply means the addition of a phosphate group to a substance. Hydrogen ions are also added to 3PG from NADPH, which, as it drops off the hydrogen ions, turns back into NADP. The G3P formed is then used to synthesize glucose, or can be converted back into RUBP to allow the cycle to continue. The glucose that has been synthesized from the G3P can be used for respiration, or can be stored as starch. It may even be used for the production of cellulose for cell walls, or can act as building blocks for molecules like fats or proteins. That is it. Photosynthesis explained at the molecular level in under 3 minutes. By no means are you obligated to do so, but a like would be much appreciated if you have enjoyed the video. If you have any video suggestions for what should be next in the Explained in 3 Minutes series, pop them into the comments. Thanks for watching.